In this class, I will discuss about T cell differentiation and these effector functions. But before you know the different subsets of T cells and effector functions, you must know one thing is called polarizing cytokine. Polarizing cytokine. And depending on the cytokine, different subsets can be produced. So this is called polarizing cytokines that actually determine the subset of T cells. So this polarizing cytokine determine subset T helper cells, and these T helper cells actually, this subset of T helper cells express a master regulatory molecule. So master regulator, this is actually a transcription factor. Subset secrete a particular cytokine and activate particular immune cells and perform its last thing, that is effector function. So the first polarizing cytokine is IFN-gamma, IL-12, IL-18. When these cytokines are secreted by dendritic cell, macrophage, NK cells, when these three cells sense pathogen, they secrete this polarizing cytokine. T helper cell, naive T helper cell become TH1 subset. A master regulator starts to express for these interactions, that is called, I am mentioning in red color, you must remember the master regulator is a transcription factor that actually determines the fate of this T helper cell. This is T beta. Okay, and a cytokine that is secreted by this TH1, because to perform helper cell to perform its function, it must secrete some cytokines, that is IFN-gamma. So here you can see IFN-gamma is activating a naive T cell into TH1 that is also secreting IFN-gamma, and this TH1 cells particularly activate macrophage, those macrophages that are actually engulfed or phagocytosed intracellular bacteria. So the effector function is to activate macrophage that engulf intracellular bacteria. The second cytokine is IL-4, interleukin 4. This cytokine can be secreted by basophil, mast cell, even follicular helper cells. These three cells can secrete. This basophil and mast cells secrete IL-4 in response of helminth. So the effector function of this subset to eliminate helminth, I can write it here, helminth. In response of helminth, this basophil cell, mast cell and T follicular helper cell can secrete IL-4, and the naive TH cells become TH2 subset. The important or master regulator for these cells is GATA3, and the cytokine that actually expressed or secreted by this TH2 cell is IL-4, IL-5 and IL-13. These are three important cytokines secreted by this TH2 cell. These cells, actually TH2 cells, can activate B cell directly, eosinophil directly, mast cell and basophil cell indirectly. What happens when this TH2 cell activate B cells with the help of IL-4, specifically I am telling you IL-4 cytokine? This B cell is activated by IL-4. This B cell starts to secrete IgE antibody. It's class switch, it's antibody to IgE type. Now this IgE can activate these two cells, basophil and mast cells. If cytokines like IL-1, IL-6, IL-23 is secreted, TGF-beta, if these cytokines are secreted, then T helper 17 is generated from the naive T helper cells, and the important or major master regulatory transcription factor is ROR-gamma T, ROR-gamma T. This is a transcription factor. The cells express or secrete cytokine like IL-17 and IL-22. These are the two major important cytokines secreted by these cells, and the cells actually activate a major important cell called neutrophil. There are many functions performed by T helper cells, that is major is inflammation. So I can write inflammation. Ultimate result, effector function of these cells is inflammations. That not only inflammations, it actually helps to eliminate extracellular bacteria, some bacteria are extracellular, and fungi. Now there is another cell that could be activated by IL-2 and TGF-beta, that is called, this is another subset called iTreg. The naive TH cell can be converted into I T regulatory cells. This is a T regulatory cell, but not actually natural T regulatory cell. It is induced T regulatory cell. So it is called induced T regulatory cell. And the master transcription factor is FOXP3. Actually, I am working on these T cells in case of tumor induced T regulatory cells, and this cell can secrete two major important cytokines, that is called IL-10 and also TGF-beta. Now this IT regulatory cell is not activate, do not activate immune cells, it inactivates. So I have to write here inactivate, it not activate, it inactivate all immune cells. So it is actually suppressive cells. So the main function of this iTreg or induced Treg is to suppress IL-6 and IL-21. There is another subset of T cells called T follicular helper cell, and the master regulator of this cell is BCL6, and this cell can secrete IL-4 and IL-21 major. They can secrete and activate, this cell activate B cells. This is called follicular helper cells, follicle, and we know that in the secondary lymphoid organ like lymph and spleen there is follicle where B cells reside. So these cells actually activate B cells and helps to form germinal center, and in the germinal center two major important thing occurs. You must remember, I will take another separate class on this T follicular helper cells, how activate B cells. Two event, one is affinity maturation by a process called somatic hypermutations. Another thing is class switching. By the help of T follicular helper cell, B cell undergo two major important events, one is affinity maturation and class switching.